Good day, Miss Majolison. I am Tugonon Blanche Abegaile from Bsed Science 2B-DAY, and I will going to explain my network diagram, which are all about the chemical nature of enzymes, the properties of enzymes, the classification of enzymes, the function of enzymes, and of course, we have the nomenclature and activities of enzymes. Let's start with the chemical nature of enzymes. So for the chemical nature of enzymes, enzymes can be denatured and precipitated with salts, solvents, and other reagents. This entire active complex is referred to as the hollow enzyme, which includes the apoenzyme or the protein portion, plus the cofactor or the coenzyme, prosthetic group or metal ion activator, which is called the hollow enzyme. So according to Holm, the cofactor may be number 1, a coenzyme, number 2, a prosthetic group, and number 3, a metal ion activator. And now, let me proceed to the three properties of enzymes, which are the physical properties, the chemical properties, and the general properties. For the physical properties of enzymes, physically enzymes behave as colloids or as substance of high molecular weight. Enzymes are destroyed or inactivated at temperature below the boiling point of water. At 60 degrees Celsius, most enzymes in liquid medium are inactivated. For the chemical properties of enzymes, we have the catalytic properties, in which enzymes are biological catalysts. The small quantity of enzymes catalyzes the larger quantities of substances. And we also have the specificity of enzymes, in which enzymes are highly specific in nature, particularly a particular enzyme can catalyze a particular reaction. For example, enzyme sucrase can catalyze only hydrolysis of sucrose. For the general properties of enzymes, enzymes initiate and accelerate the rate of biochemical reaction. The activity of enzymes depends upon the acidity of medium or the pH specific. Enzymes can accelerate the reaction in either direction. And now, let me proceed to the classifications of enzymes. Enzymes can be classified into 6 major groups according to the type of reaction they catalyzed. We have number 1, oxidoreductases, which catalyze oxidations and reductions. Number 2 is the transferases that catalyze the transfer of a group of atoms, such as from one molecule to another. Number 3, we have the hydrolases that catalyze hydrolysis reactions. Number 4, we have the lyases that catalyze the addition of two groups to a double bond or the removal of two groups from adjacent atoms to create a double bond. Number 5, we have the isomerases that catalyze isomerization reactions. And lastly, we have the ligases or the synthetases that catalyze the joining of two molecules. And now, let me proceed to the functions of enzymes. The enzymes perform several functions in our bodies, and these include enzymes help in signal transduction, in a way that the most common enzyme used in the process includes protein kinase that catalyzes the phosphorylation of proteins. Number 2 is that they break down large molecules into smaller substances that can be easily absorbed by the body. Number 3 is that they help in generating energy in the body in a way that ATP synthase is the enzyme involved in the synthesis of energy. Number 4 is that enzymes are responsible for the movement of ions across the plasma membrane. Number 5 is that enzymes perform several biochemical reactions, including oxidation, reduction, hydrolysis, and etc. to eliminate the non-nutritive substances from the body. And lastly, they function to reorganize the internal structure of the cell to regulate cellular activities. Now, let me proceed to the nomenclature of enzymes. The nomenclature of enzymes is derived from their substrates or the catalyzed chemical reactions, and ASE is usually added as a suffix. Nearly all enzymes end with the suffix of ASE. Generally, the names are of the form substrate or product in which the reaction catalyzed. So lastly, we have the activities of the enzyme. So we have here the so-called lock and key model, the induced fit model, and the enzyme inhibition. So lock and key model was proposed by Emil Fischer in 1894. So according to the model, the shape of the substrate and the active site of the enzyme are thought to fit together. While the induced fit model was introduced or was proposed by Daniel E. Koshland Jr. in 1958. It states that the binding of substrate induces a conformational change in the active site of the enzyme. In addition, the enzyme may distort the substrate forcing it into a conformation similar to that of the transition state, like a key into its lock. So lastly is the enzyme inhibition that consists of first is the activation in which it is any process that initiates or increases the action of an enzyme. It can be the simple addition of a cofactor to an apoenzyme or the cleavage of a polypeptide chain. It is a cleavage of a polypeptide chain of an enzyme, and second is the inhibition, which is the opposite or any process that makes an active enzyme less active or inactive. So inhibitors are compounds that accomplish this task, and there are many types of enzyme inhibition. So third is the competitive inhibitors, which bind to the active site of the enzyme surface, thereby preventing the binding of substrate. Lastly are the non-competitive inhibitors that binds to some other portion of the enzyme surface and sufficiently alter the tertiary structure of the enzyme so that its catalytic effectiveness is eliminated. That is, the enzyme cannot catalyze while the inhibitor is bound. Both competitive and non-competitive inhibition are reversible, but some compounds alter the structure of the enzyme permanently and thus make it irreversibly inactive. So that would be all, Miss Majlison. Thank you and God bless.